What's going on? My name is Mr. Chipman. I teach biology and AP biology at Murray High School in Murray, Kentucky. And here we are continuing our study in Unit 1, looking at 1.5 lipids. Looked at carbohydrates in 1.4. Hopefully you're able to watch that. Now we're going to look at lipids. Lipids are important things. Here we go. Lipids. So lipids are fats, right? Just because they're fats, well, not just because they're fats, but lipids are nonpolar. Another way you can say this is that they are hydrophobic and they do not dissolve in water. We talked about water's polarity, right? We said because water's polar, water's able to dissolve things that are also polar, universal solvency. We talked about this idea, right? And a nonpolar molecule doesn't have any unequal sharing. Everything is like equal. It's almost like trying to grab a hold of something that's really smooth, right? There's no unevenness to it, and so there's no way to like grab it. Polar molecule has some unevenness, right? And so they're able to grab each other, but nonpolar things aren't. And so polar things and nonpolar things don't like each other. And so you'll see those two words, hydrophobic, hydrophilic, which is water loving, very commonly used, and all lipids are hydrophobic. Fats, oils, phospholipids, we're going to talk about steroids, waxes, also important. Main function for lipids is energy storage, so more long-term, but we see other uses for lipids as well. Insulation for particularly mammals, protection for vital organs, cell membrane structure. We're going to get into this. So let's talk about the structure. So a lipid, typically, not always, but just generally, is made up of these fatty acids, which are considered the monomer for lipids. And these fatty acids typically can be seen in threes. This is a triglyceride. You've probably heard that term, right? It's very commonly used in like blood work or whatever. So triglyceride, 3 fatty acids hooked to this glycerol. I like to think of the glycerol sort of like, kind of like a closet rod. This worked for me. Like a closet rod, and you're like putting your hangers on the clothes and you hang them up and you have 3 of them. Those are your fatty acids. The closet rod kind of keeps it all together, right? Now, a couple of things about lipids that you need to be aware of. Lipids are primarily made of carbon and hydrogen. Sometimes you'll hear them called hydrocarbons, right? Not carbohydrates. The hydrate is water, right? Hydrogen, carbon. There is oxygen, just a little bit of oxygen here, but mostly just long chains of carbon and oxygen, or carbon and hydrogen. Remember we talked about with carbohydrates, we said the carbons and oxygens are going to be in equal proportion. They are not in lipids. Carbons and hydrogens by far and away outnumber the oxygens that are available in a lipid. And so that's a way to recognize a lipid. These long chains, there are a couple of lipids that are going to be very specifically structured that we'll look at, but most lipids are going to have this sort of lengthy appearance to them. They may be kinked, which we're going to look at. So very important. Speaking of kinks, fatty acid, some of them are straight, nice and straight, kind of like a pencil, right? Imagine grabbing a brand new handful of pencils, like unsharpened wooden pencils. They kind of form this like structure, like really nice and tight structure, right? Because they're all sort of interlocking-ish, like hexes, right? Saturated fats are like that. They're nice straight lines. They interlock really easily. That's why they form solids at room temperatures, because they're just nice and solid. Unsaturated fats don't do that. They're bent, so they don't form solids. They form liquids at room temperature. And why are they bent? It's because these carbon double bonds here. Notice everything's kind of full. Could you put anything else in here? No. What's it full of? It's full of hydrogen, right? You couldn't put anything else in there. What about here? Yeah, there's some space for some more hydrogens. Of course, you had to break these bonds. You had to cram them in there artificially, which we'll talk about. But an unsaturated fat in natural form is bent, and so it's a liquid. This is like vegetable oils. Typically, saturated fats are like animal oils, but those two rules are not always hard and fast, so be careful there. Saturated fat is completely saturated or full of hydrogen. Unsaturated fat is not because of the double bonds. Triglyceride, we talked about triglyceride already. Just to show you, it's another picture of it. Again, how are the fatty acids joined to the glycerol? That is in dehydration synthesis. If you want to break them off, so hydrolysis, very important. Here's a phospholipid. phospholipid. Think phospho. What does it sound like? Phosphorus. It's a real important thing here. This phosphate, we're going to talk a lot about, you're going to hear the word phosphate a lot this year. Phosphate is not hydrophobic. It's hydrophilic. It's got a charge to it. Loves water, in fact. Just loves it. These chains do not. And so a phospholipid is actually a part of a cell membrane that has two charges. Has a hydrophilic head and a hydrophobic tail. We will talk more about that when we get to that unit. But for now, it's an important lipid. Makes up cell membranes and our cell plasma membranes, and very, very important structure. I'm going to go back up here and talk about this because I don't think I have a slide for it. I mentioned that if you could cram some hydrogens into this unsaturated fat, sometimes you'll see those called hydrogenated fats. It's just artificially been crammed full of hydrogen. This is like Twinkie cream and Cool Whip. Sorry, I didn't mean to spoil Thanksgiving. So steroids. Steroids are a type of lipid. Interesting thing about steroids. So look here. Steroids are always going to have this sort of ring-like structure, 4 rings, and then some sort of additional thing. What's going to make cholesterol different than cortisol? Well, it's this additional thing because shape determines function, right? Good. And so steroids are chemical messengers. Interesting thing about a steroid is that, we're going to get into this when we get into talk about Unit 4, I think, is a steroid can pass directly through the cell membrane. Why is that? Well, it's that nonpolar thing, right? It's like they like wink at each other when they go by. It's like, it's like the bouncer at the club door. Somebody knows them, they get in. It's a bad illustration, maybe. Okay, steroids, chemical messengers, very important. Waxes. Now, interesting thing about wax. I read a lot about waxes when I was doing this because I did, it's different. Waxes are a very normal, natural thing. They're composed of these long fatty acid chains. These R's represent those long chains, right? Anytime you see that, it's kind of short for a chain. But all waxes are going to have this sort of structure right here, which is this long chain that is linked to a chemical called an alcohol. And when you hear alcohol, you think drinking alcohol. But alcohol is a type of chemical too, and it can take on different forms. And the purpose of a wax is, you can see on this plant, provides protection from what? Well, from water. If a plant just is saturated with water, what's going to happen to this leaf? It's going to break off, right? So the leaf kind of lets the water kind of skirt down to the ground. Where does it want the water? Well, it wants the water in the ground anyway, right? And so the wax actually forms what's called a cuticle on the outside of the plant, which allows water to be repelled. Waxes are used in other features in animals and plants as well that we're not going to get into, but very important. Earwax is a great example of that. We don't want water in our ears. That never works out. A couple of other things to talk about with lipids. Energy storage. Why are they really good at storing energy? Because they have tons of bonds. Right? It's different than a glucose. But glucose is good. It's a good source of energy. But it can't beat fats in the amount of energy that it has. Right? Fats have a ton of energy. But think about if you had a gram of glucose versus a gram of like oil. Which one of those is going to burn longer and hotter? Right? There's more energy in that oil for sure. And so our body uses this, and not just our bodies, but all organisms use this to their advantage by storing fat in order to store energy long term. If you have more energy than you need, you store it long term as fat. There's different kinds of fats. Cell membranes, obviously, we talked about. The important thing about cell membranes, we'll get more about this when we get into that unit, is that it forms a protective barrier. That lipid performs a protective barrier around the cell, protects it from things that shouldn't be going in. Of course, steroids, they get in for free. System regulation, we talked about this with like glycolipids, some other sort of steroids, cellular communications, very important function there. Steroids are very important for body function. A lot of reproductive hormones are steroid hormones, so very important for body function. And one more thing, insulation and protection. So again, insulation keeps us warm. There's a certain level of fat that's necessary in order to sort of keep us warm from freezing in colder temperatures. And then storage of fat for, if you have more energy than you need. And then also to protect our vital organs. Fat is used to cushion vital organs in order to keep them from breaking when you fall or when you jump or whatever it is that you're doing. It protects those organs. Here's a question. Let's look at a question. Pause the video if it helps. And we're going to go over the question in 3, 2, 1. Here we go. Student compares two artificial membranes. Membrane A contain mostly saturated fats, and membrane B unsaturated fats. Student observes that membrane B remains more fluid at low temperatures than membrane A. Well, that's shocking. Which of the following best explains this difference in membrane fluidity? Pause the video again if you need to do that, but we know why. Saturated fats are straight. There's not bent, right? And unsaturated fats are bent, and so it allows them to, or doesn't allow them to line up, and so they stay a little more fluid. We're going to go find that now in the answers. We came up with something before we went to ABCD. We're not going to let ABCD confuse us, right? We're going to go find the answer now. Double bonded. And double bonds in saturated fats, that's already wrong. I'm done. There aren't double bonds in saturated fats, right? Single bonds in saturated fats allow tighter packing, reducing membrane flexibility. I like that one. It's not wrong, but I'm a little unsure, right? So let's look at C. Unsaturated fatty acids have more polar regions. Yeah. Unsaturated fatty acids increase membrane permeability by creating pores. What? We should have went with B before reading C and D because C and D were so silly. B is the correct answer. Yeah, makes it less fluid. Or, for me, what I was doing, why did I go ahead and read C and D? I'll just be honest with you. I was like, well, I'm really looking forward to describe unsaturated fats, but it kind of went the backwards way. You may have been fine with B, but, sometimes you just need to read C and D as well. Hopefully this was helpful. If it was, let me know. If you have questions, let me know down in the comments. Thanks for watching. We're going to be moving on to 1.6 pretty soon.